You begin by doing something very simple. You slow down. Not just your breath, but your attention. The world outside the room softens, its sharp edges rounding off, and your awareness drifts inward, toward the steady, unnoticed rhythm of your own body. Nothing dramatic happens. There is no sudden revelation. There is only a quiet hum, constant and reassuring, like a distant engine running smoothly through the night. Inside you, trillions of tiny lives are awake. Each cell is a small room with its own gentle order. Walls made of membrane hold everything in place. Fluids move slowly, carrying nutrients like warm currents. Proteins fold and unfold with practiced ease. This is not chaos. It is choreography, refined over billions of years. You imagine yourself floating closer, shrinking down until you can drift through these rooms without disturbing them. No alarms sound. The cells do not notice you watching. They are too busy being alive. In the center of most of these rooms lies something precious and quiet, DNA. It is not loud or glowing. It does not demand attention. It simply exists, coiled softly like a sleeping ribbon. DNA is often drawn as a twisted ladder, but here it feels more like a long, handwritten letter. The ink is chemical, the paper molecular, but the idea is familiar. Instructions, passed carefully from generation to generation, telling each cell how to be what it is. How to become skin or bone, blood or thought. How to repair itself. How to rest. For most of human history, this letter remains sealed. People sensed that something inside the body carried information, but they could not see it clearly. They listened instead to symptoms, to inheritance, to patterns repeating through families like half-remembered songs. They learned patience before they learned precision. Listening is an underrated skill in science. It requires stillness. It requires admitting how much you do not yet understand. Early biologists did not rush in with tools. They observed. They noticed that traits traveled from parent to child. That tiny changes could echo across generations. That life remembered itself in ways no one could yet explain. As technology improved, listening became sharper. Microscopes opened new doors. Stains and dyes revealed hidden structures. Gradually, the letter inside the cell became legible. Not readable in words, but in patterns. Sequences. Repetition and variation. The alphabet of life began to emerge, not as poetry, but as chemistry. You rest with that idea for a moment. That before editing anything, humanity spent decades simply learning the language. For chemical letters repeating endlessly, arranged in different orders, forming instructions long enough to fill libraries. Every cell in your body carries a copy. Not because it needs all of it, but because the story is shared. There is comfort in that sharing. The same basic code appears in trees, in birds, in bacteria drifting through soil and sea. Differences exist, of course, but the foundation is familiar. Life, it turns out, prefers to reuse what works. Evolution edits slowly, patiently, over time spans too long for urgency. As scientists learn to read DNA, they also learned restraint. Reading is not the same as touching. Understanding does not require interference. For a long while, the goal was simply to know where genes were, what they influenced, how they interacted. The body was treated like a book in a rare collection. Observed. Respected. Left intact. You imagine researchers late at night, labs dim and quiet, machines humming softly. Data appears slowly, line by line. Discoveries arrive not as shouts, but as whispers. A gene linked to a color. Another to blood type. Patterns forming gently, like constellations drawn between distant stars. This period of listening changed how science thought about life. It revealed that most of DNA is not about immediate action. Much of it regulates, pauses, or waits. Some sections seem silent at first glance, yet they shape when and how other instructions speak. Silence, it turns out, is part of the message. You feel your own body breathing, cells responding automatically. No one is directing them consciously. The instructions run quietly in the background, stable and reliable. This is what made scientists cautious. Editing such a system would never be simple. Every change might ripple outward, touching places unseen. So before tools were built, questions were asked. What does this gene do? What happens when it changes naturally? Which variations are harmless, and which matter deeply? Listening meant tracking consequences across time, not moments. It meant humility in the face of complexity. Gradually, the idea of precision began to form. If life already edits itself slowly through mutation and repair, could humans one day guide that process? Not randomly, not forcefully, but carefully, the way a proofreader marks a page, aware that every correction changes the meaning of the sentence. You sense the shift happening here, in this quiet chapter of the story. The move from observer to participant has not yet occurred, but it is approaching. The listening has trained the ear. The language feels less foreign now. Letters repeat. Patterns stabilize. The once mysterious letter inside the cell becomes something you can hold in your mind. Still, no scissors appear yet. No edits are made. The night remains calm. Science sits beside life, not above it, learning its rhythms. This is the necessary beginning. Attention before action. Understanding before intervention. As you rest with this awareness, the DNA inside you continues its silent work, unchanged, patient. And somewhere in that vast, shared story, the idea begins to form that if the letters can be read, perhaps one day they can be followed, traced, and understood line by line, as the long story written in DNA slowly comes into focus. You continue inward, carried by the same quiet attention, and now the DNA no longer feels distant or abstract. It feels present, intimate, like something that has always been with you. Inside each cell, the long molecular thread rests in careful coils, folded not from disorder, but from necessity. Space is limited, and the story is long. You imagine unrolling it slowly, not all at once, but section by section. The DNA does not rush to reveal itself. It has learned patience over billions of years. What you see is not a single instruction, but a vast sequence of letters, arranged with calm consistency. For chemical symbols repeat endlessly, like a simple alphabet used to write an infinite number of sentences. These letters are older than any spoken language. Older than writing carved into stone. They existed before eyes could see them, before minds could wonder what they meant. Yet they carry meaning all the same. Not symbolic meaning, but functional meaning. When read by the cell, they guide action. They tell proteins when to form, where to go, and when to rest. You sense that this story is not read from beginning to end like a novel. It is more like a reference library. Different sections are opened at different times, depending on need. A skin cell reads one set of instructions. A neuron reads another. The same book, interpreted in many ways, shaping diversity from sameness. As you linger here, the idea of mutations gently enters your awareness. They are often described as mistakes, but that word feels too harsh. In this quiet place, mutations feel more like typos made by time. A letter copied slightly differently. A phrase repeated or skipped. Most of the time, these changes do very little. The sentence still makes sense. The story continues. Sometimes, though, a small change alters the meaning. A protein folds differently. A signal weakens or strengthens. Over generations, these changes accumulate, slowly editing the story without intention. Evolution works like a patient editor who never sleeps, reading drafts across millions of years. You realize that without these typos, life would not adapt. Variation is not a flaw. It is a feature. The long story written in DNA survives because it allows small differences to explore new possibilities. Some changes fade quietly. Others persist, shaping bodies, behaviors, and entire species. Scientists learn this gradually, by comparing stories. They place DNA side by side, noticing similarities and differences. Human and mouse. Tree and fungus. Bacterium and whale. The shared passages were striking. The differences told their own subtle tales of divergence and survival. This comparative listening revealed something profound. The story is not static. It is alive. Not alive in the way a creature is alive, but alive in its responsiveness. DNA responds to pressure, to environment, to chance. It bends without breaking, preserving the core while allowing variation at the edges. You rest with that thought. That your own DNA carries echoes of ancient environments. Cold nights. Scarcity. Abundance. Movement. Stillness. These experiences are not memories in the human sense, but traces, encoded quietly in molecular patterns that once helped ancestors survive. Understanding this long story required humility. Early scientists hoped for simple answers. A single gene for a single trait. Sometimes that was true. Often, it was not. Traits emerged from conversations between many genes, influenced by timing and context. The story was layered, recursive, and subtle. This complexity slowed progress, but it also deepened respect. Editing such a story would never be like crossing out a word with a pen. It would require understanding how sentences relate to paragraphs, how chapters influence the whole. Listening became more refined, more cautious. You imagine researchers learning to map the genome, charting its vast landscape. Peaks of activity, valleys of silence, regions that regulate rather than instruct. DNA was no longer seen as a rigid blueprint, but as a dynamic script, responsive and adaptable. In this light, mutations lose their moral weight. They are not good or bad. They are changes. Context decides their impact. A typo that matters in one environment may be harmless in another. The story adapts not by perfection, but by flexibility. You feel the comfort in that idea. That life does not demand flawlessness. It tolerates variation. It learns through it. Your own body carries countless tiny differences, most of which you will never notice, all coexisting peacefully as the story continues to be read. As night deepens, the letters feel less mysterious. Familiarity replaces awe, not by diminishing wonder, but by softening it. You are not outside the story, looking in. You are written into it. Every breath you take is guided by lines composed long before you were born. And as understanding settles in, another thought begins to form. If this story can be read, compared, and mapped, then somewhere ahead lies the question of memory. How life remembers past threats, past encounters, past lessons, storing them quietly, waiting for the right moment to be recalled. You drift further back now, not just inward, but backward in time. Long before humans learned to read DNA, long before bodies became complex, life was already practicing survival. The world was simpler, harsher, and filled with uncertainty. In ancient waters, tiny bacteria floated freely, unaware of names or futures, yet deeply aware of danger. These bacteria lived moment to moment. They absorbed nutrients. They divided. They responded. And sometimes, without warning, they were attacked. Viruses slipped through their boundaries, hijacking their inner machinery, turning life into a factory for something else. Most bacteria did not survive these encounters. But some did. You imagine one of them drifting calmly after the danger passes. Inside this tiny cell, something unusual happens. A small fragment of the invading virus is saved. Not out of malice. Not out of fear. Simply as a record. A molecular snapshot of what nearly ended its existence. This is memory at its most basic. No awareness. No emotion. Just information stored quietly for later use. The bacterium continues living, dividing, passing this fragment along to its descendants like a folded note tucked safely away. Over time, this practice becomes refined. Bacteria that remember survive more often. Those that do not, fade. Nature does not reward intention, only outcome. Slowly, a system takes shape. A library of fragments, each one representing a past threat. Each one held patiently, waiting. You sense how different this memory is from your own. There are no images, no feelings. Yet it is effective. When the same virus returns, the bacterium recognizes it instantly. The stored fragment acts like a quiet alarm, guiding defensive action with remarkable precision. This system comes to be known as CRISPR, though the bacteria never name it. For them, it is simply part of living. Clusters of repeated DNA, spaced by fragments of past invaders, arranged carefully along the genome. A record of survival written directly into the long story of DNA. You rest with that image. Memory embedded not in a brain, but in the code itself. Experience becoming instruction. Past danger shaping future response. This is not learning as humans understand it, but it is learning all the same. For centuries, this system goes unnoticed. Bacteria continue their quiet lives beneath awareness. Humans see them only as nuisances, or not at all. The memory they carry remains hidden, working silently in soil, water, and bodies. Then, much later, scientists begin to listen more closely. They notice strange repeating patterns in bacterial DNA. Sequences that do not seem to code for proteins. Sections that appear again and again, separated by unfamiliar fragments. At first, these patterns are puzzling. They do not fit existing explanations. So scientists do what listening requires. They wait. They compare. They observe. Gradually, the truth emerges. These fragments match viral DNA. The bacteria are not just reacting. They are remembering. They are keeping a molecular diary of past attacks. You can feel the shift this realization creates. DNA is not only a story of inheritance. It is also a story of experience. Life does not merely pass instructions forward. It records what it encounters, editing its own defenses over time. This discovery changes how humans think about control and intention. The bacteria did not design this system. It emerged because it worked. Precision arose from patience. Defense from repetition. Nature, it seems, had already solved a problem humans had not yet asked. As scientists understand this system more deeply, they see its elegance. When a familiar virus appears, the bacterium produces small guide molecules copied from its stored fragments. These guides move through the cell, searching. When they find a matching viral sequence, they bring with them a cutting enzyme. The threat is neutralized quickly, efficiently, without panic. You notice how calm this process feels. There is no struggle. No waste. Just recognition followed by action. Memory guiding response with quiet certainty. This is the moment where listening turns into possibility. Humans realize that this bacterial memory system is programmable. That fragments can be added, removed, redirected. That the same tools bacteria use to defend themselves could, one day, be guided elsewhere. But that thought remains gentle, distant. For now, the wonder lies in understanding. In seeing how something so small carries such sophistication. How survival can produce systems of breathtaking clarity without conscious design. You breathe slowly, imagining these bacteria continuing their ancient work all around you. In your gut. In the soil beneath your feet. In oceans still echoing with the earliest forms of life. They remember. They adapt. They persist. And as your awareness lingers on this quiet intelligence, you sense the next step approaching. If bacteria can remember and recognize with such precision, then the tools they use to act on that memory must be equally careful, equally exact. You stay with the image of bacterial memory, and slowly your attention shifts from remembering to acting. Memory alone is gentle, but survival requires response. Somewhere inside the cell, a decision must be carried out. Not loudly, not aggressively, just accurately. In this tiny world, action takes the form of proteins. They are not rigid tools, but soft, folded shapes, drifting through the cell like careful hands. Each protein is built from long chains of amino acids, folded again and again until form becomes function. Shape is everything here. A slight curve determines what can be held, what can be cut, what can be ignored. Among these proteins is one that feels different. Calm. Focused. It does not move randomly. It waits. This is the Cas enzyme, the molecular scissor at the heart of CRISPR. It does not search on its own. It does not decide. It listens. You imagine it floating in the cell's interior, surrounded by motion, yet unmoved by it. Nutrients pass. Signals ripple. Still, the enzyme remains inactive, like a tool laid gently on a table, untouched until needed. What gives this scissor its power is not sharpness alone, but restraint. Left unguided, it does nothing. It requires instruction. It requires a guide. Without one, it remains quiet, folded into readiness, refusing to act on chance. This is an important moment in the story. For all the fear humans sometimes feel about cutting DNA, nature has already built in hesitation. Action is never automatic. Precision is valued more than speed. When the guide finally arrives, copied from a remembered fragment, everything changes slowly, deliberately. The guide molecule binds to the Cas enzyme, fitting into it like a key into a lock. Now the scissor knows what to look for. Not vaguely. Specifically. Together, they move through the cell, scanning the long strands of DNA. This search is not rushed. The guide checks each sequence patiently, letter by letter, testing for a match. Most of the time, it moves on. Near misses are ignored. Similar is not enough. Precision is required. You sense how careful this process is. The enzyme does not slash wildly. It pauses constantly, confirming identity. Only when the guide finds a perfect or near-perfect match does the enzyme prepare to act. And when it does, the cut is clean. The DNA is sliced at a precise location, both strands severed in a controlled motion. There is no tearing, no chaos. Just a momentary break, created exactly where intended. The threat is neutralized. The virus is stopped. You rest with this image, noticing how different it feels from human tools. There is no excess. No collateral damage. The enzyme does not linger. It does not cut again unless guided again. Power here is conditional, earned through accuracy. When humans first understand this system, there is a quiet astonishment. Not because cutting DNA is possible, but because it is done so thoughtfully. Nature has created a tool that waits to be told what to do, and refuses to act otherwise. This realization changes everything. If such scissors exist, then perhaps DNA is not untouchable. Perhaps it can be edited, not violently, but surgically. With the same care bacteria used to defend themselves. But the responsibility is heavy. Cutting DNA is not trivial. The genome is not just a string of letters. It is a living context. Every cut sends ripples. Cells notice. Repair systems activate. Consequences unfold. Scientists learn quickly that the Cas enzyme does not finish the story. It only creates an opening. What happens next is up to the cell. You imagine the cell responding calmly to the break. It has encountered damage before. Breaks happen naturally, from radiation, from chemicals, from the act of living itself. Over time, cells have evolved ways to fix these breaks, to restore continuity. So the cut made by the protein scissor is not an ending. It is a question posed to the cell. How will you repair this? This is why precision matters so deeply. Where the cut is made determines what the cell does next. A break in one place may silence a gene. In another, it may invite change. The same scissor, used differently, produces different futures. You feel the weight of this realization. Tools do not carry intention. They carry possibility. Intention comes from guidance, from choice, from understanding context. In bacteria, this choice is shaped by survival alone. In humans, it is shaped by ethics, curiosity, and care. The same molecular scissor, now held in human hands, demands more than technical skill. It demands wisdom. As night settles deeper, the enzyme drifts back into stillness in your imagination. Its work is done. The DNA, momentarily opened, waits for repair. The system remains balanced, quiet, alert. You sense that this chapter is not about cutting, but about hesitation. About the value of waiting until you are certain. About building tools that do nothing until guided with clarity. And in that calm pause, another idea begins to emerge. If scissors are only as good as their instructions, then the next question is not about cutting at all, but about teaching molecules where to look. You remain close to the moment after the cut, where stillness returns and attention shifts. The scissors have done their work, but they do not decide when to move again. That choice belongs elsewhere. In this calm molecular landscape, action always waits for guidance. You begin to notice something smaller now, lighter, moving with purpose but without weight. It drifts through the cell like a thought forming slowly. This is the guide RNA, though here it feels less like a molecule and more like an intention made visible. It is not strong. It does not cut. Yet nothing meaningful happens without it. Guide RNA is born from memory. It is copied from the fragments stored long ago, the pieces of viral DNA preserved like pressed leaves between pages. These fragments are transformed into a form that can travel, that can search. They carry information, not as explanation, but as pattern. You imagine this guide as a soft ribbon, flexible and alert. It does not scan the DNA aggressively. It glides along it, touching gently, testing connections. Letter meets letter. Most do not match. The guide moves on without frustration. There is no urgency here, only patience. This patience is essential. DNA is vast. Even in a simple cell, it stretches long and complex. Mistakes would be costly. So the guide checks carefully, verifying identity before signaling action. Near matches are not enough. Similarity is ignored. Only alignment earns response. You sense how different this feels from human searching. There is no anticipation, no hope. Just process. Recognition is either present or it is not. When it is not, the guide continues drifting, content to wait as long as needed. When the match finally appears, it feels quiet rather than dramatic. The guide settles gently against the DNA, pairing with it perfectly. This moment of alignment is the true decision point. Not the cut, but the recognition. Only now does the Cas enzyme respond. It does not rush in blindly. It binds to the guide, confirming that instruction is present. The guide does not command. It suggests. Together, they form a partnership built on agreement. You rest with the idea that the most powerful step in this process is informational. Before force, there is direction. Before action, there is certainty. The system is designed to minimize regret. When humans adapt this system, this lesson becomes central. Editing DNA is not about stronger scissors. It is about better guidance. Scientists spend far more time designing guide RNA than wielding enzymes. The guide determines everything that follows. Designing a guide requires deep understanding. The genome must be mapped. Variations must be considered. Similar sequences elsewhere must be avoided. Each guide is chosen with care, tested again and again, refined patiently. You imagine researchers sitting quietly with screens glowing softly, comparing sequences, searching for uniqueness. Their work mirrors the guide itself. Slow, careful, focused. They are teaching molecules where to look, not how to cut. This process reshapes how science thinks about control. Power becomes distributed. The enzyme holds strength, but the guide holds intention. Neither is sufficient alone. Only together do they act. You notice how this reflects something deeply human. Words guide actions. Maps guide journeys. Instructions guide tools. Without direction, strength is wasted. Without clarity, effort becomes harm. In the cell, the guide RNA eventually releases its match. Its work is temporary. It does not linger or attach permanently. Once recognition and cutting occur, it drifts away, dissolving back into the molecular crowd. Its job is done. This impermanence matters. Nothing here claims ownership. Each step exists only as long as needed. The system values efficiency and restraint over dominance. As the night deepens, you feel the rhythm of this process settling into your own breathing. Search. Pause. Recognize. Act. Release. It is a cycle that feels calming rather than mechanical. You begin to see how this gentle guidance makes editing possible at all. Without it, scissors would be reckless. With it, they become precise instruments of change. Not perfect, but thoughtful. There are still limits, of course. Guides can sometimes misread. Similar sequences can confuse. Scientists work constantly to improve accuracy, learning from errors the same way cells do. Listening never ends. You sense that the story is shifting again. Guidance has led to cutting, but cutting is only an invitation. The cell itself now prepares to respond, to mend what has been opened. The guide has done its work. The scissors have paused. The DNA waits, open and quiet, trusting the next step. You remain with the DNA as it rests in its open state, not broken in fear, but paused in possibility. The cut has already happened, precise and deliberate, and now there is a brief silence. In this silence, nothing feels ruined. Instead, the cell recognizes something familiar. Breaks are not strangers here. They are part of life. Every day, even without guidance or intention, DNA experiences small injuries. Heat, radiation, chemistry, and motion all leave their marks. Over billions of years, cells have learned not to fear these moments. They have learned to respond. So now, as the strands part, repair systems awaken gently. Proteins drift toward the opening like skilled hands approaching torn fabric. They do not rush. They do not guess. They assess. You imagine these repair proteins as quiet caretakers. They have seen this before. Their task is not to judge why the break exists, only to fix it in the best way available. Repair is their language. Continuity is their goal. One pathway moves quickly. It simply rejoins the broken ends, stitching the strands back together. This method values speed over precision. It restores wholeness fast, sometimes leaving small changes behind. A letter may be lost. A phrase may shorten. Often, the cell continues without consequence. Another pathway moves more slowly, with greater care. It uses a template, or reference, to rebuild what was cut. Missing letters are restored accurately. The original meaning is preserved. This method requires more time, more coordination, more patience. You sense the choice unfolding quietly. The cell does not weigh options the way a mind does, but conditions matter. Timing matters. Availability matters. Repair is shaped by context. This is where editing truly happens. Not at the cut, but in the repair. The scissors only ask the question. The cell answers. If the repair is imperfect, a gene may be silenced. The story shifts subtly. If a new template is provided, a corrected instruction may take its place. The sentence changes, but the paragraph continues. You feel the gentleness of this process. Editing is not an act of destruction. It is an invitation for the cell to rewrite a line in its own way. The cell remains the author. Scientists come to understand this slowly. Early excitement about cutting gives way to deeper respect for repair. Control does not lie in the blade, but in the conditions surrounding healing. You imagine researchers learning to guide not just where to cut, but how the cell might fix the cut afterward. They learn to offer templates, to influence timing, to encourage one repair pathway over another. This work is subtle and careful. Repair reveals something profound about life. Life expects damage. It plans for it. It builds systems not to prevent every break, but to recover from them gracefully. You rest with that idea. That resilience is not rigidity. It is flexibility. The ability to mend without losing identity. In this molecular world, perfection is not required. Many repairs are slightly uneven, yet life continues. Cells tolerate small imperfections. They adapt around them. Only when changes disrupt essential functions does trouble arise. This tolerance is what makes editing possible at all. If life demanded flawless copying every time, change would be impossible. Repair allows variation without collapse. You notice how this mirrors your own experience. Healing is rarely invisible. Scars form. Memories shift. Yet wholeness returns in new shapes. As the repaired DNA settles back into its familiar coil, the cell resumes its quiet rhythm. Instructions are read again. Proteins are built. The story continues, altered or preserved, depending on how repair unfolded. This is the moment where hope quietly lives. Genetic diseases often arise from small errors repeated faithfully. Repair offers a chance to soften those errors, to rewrite a line that causes harm. Yet restraint remains essential. Repair can also introduce new uncertainty. Scientists learn that not every edit produces the intended outcome. Cells are partners, not machines. You feel the humility this requires. Editing life means trusting life to respond wisely, while accepting that outcomes may vary. Precision guides the attempt. Repair determines the result. The cell does not celebrate the repair. It does not mark the moment. It simply moves on. That is its nature. Survival is quiet. As night deepens, you sense that this chapter is not about tools or techniques, but about healing. About the remarkable fact that life carries within itself the ability to mend, adapt, and continue. The scissors are gone. The guides have dissolved. What remains is the cell, whole again, changed or unchanged, but alive. And as you rest with this gentle understanding, another thought begins to surface. If repair can change a single cell's future, then across generations, careful repair might soften inherited mistakes, reshaping echoes passed down through time. You stay with the idea of echoes, letting it settle into your awareness. Some sounds fade quickly. Others linger, bouncing softly from one surface to another, repeating long after the original voice has gone quiet. In the language of biology, inherited genetic conditions are like this. They are not sudden events, but continuations. Inside families, certain patterns repeat themselves. A condition that appears in childhood. A weakness that shows up again and again. These are not punishments or flaws in character. They are instructions copied faithfully, line by line, from one generation to the next. You imagine a single letter in DNA written slightly differently. Just one. So small it feels insignificant. Yet as cells divide and bodies form, that difference becomes part of the foundation. The same altered instruction is read again and again, shaping proteins, shaping tissues, shaping lives. For most of history, there was nothing to be done about these echoes. Medicine could treat symptoms, ease pain, slow progression. But the underlying instruction remained untouched, quietly repeating itself. Listening helped. Understanding helped. But change felt out of reach. Now, with careful editing, the idea of correction begins to feel possible. Not correction in the sense of judgment, but in the sense of relief. A chance to soften a harmful instruction before it speaks too loudly. You feel how delicate this territory is. Inherited DNA is not just information. It is identity. It connects you to ancestors you will never meet. Editing it requires humility, patience, and restraint. Scientists approach this work slowly. They begin not with sweeping changes, but with specific, well-understood conditions. Disorders caused by a single, clearly identified genetic change. Places where the story is short and the sentence is clear. In these cases, CRISPR is used not to rewrite everything, but to adjust one line. The scissors cut. The cell repairs. A healthier instruction takes the place of the harmful one. The echo softens. You imagine a cell quietly reading this corrected line for the first time. It does not celebrate. It simply follows the new instruction. A protein folds properly. A signal flows smoothly. Balance returns without announcement. This is how hope appears in molecular form. Not dramatic. Not immediate. But steady. Yet even here, care dominates every step. Scientists do not rush to change future generations. Most work begins in cells outside the body, or in somatic cells that affect only the individual, not their descendants. The goal is healing, not redesign. You feel the tension that surrounds this work. The same tools that can correct harmful mutations could, in theory, be used for other changes. This awareness keeps progress slow and deliberate. Ethical questions are never far away. Still, the quiet successes matter. Cells that once struggled begin to function more normally. Tissues regain strength. Symptoms lessen. For individuals living with genetic disease, these changes are not abstract. They are deeply personal. You imagine someone breathing more easily. Someone moving without pain. Someone spared a condition they were told was inevitable. These moments do not feel like triumphs over nature, but collaborations with it. Correction, you realize, is not about perfection. It is about restoring options. Giving the body a chance to choose a healthier path. Inherited mistakes are not mistakes in the moral sense. They are outcomes of chance and history. Evolution does not plan ahead. It works with what it has. CRISPR simply allows humans to intervene gently when the cost of not intervening is suffering. You rest with the idea that this is not about erasing difference. Many genetic variations create diversity without harm. The goal is not uniformity, but relief where variation causes pain. As research continues, limits remain clear. Not every condition can be corrected. Not every edit works as intended. Cells are complex partners. Outcomes vary. Listening remains as important as acting. You sense how this careful pace protects something essential. Trust. Trust between science and society. Trust between generations. Trust that the future will not be reshaped recklessly. The idea of inheritance itself begins to shift. DNA is no longer seen as an unchangeable fate, but as a conversation across time. One that can, occasionally, be gently redirected. You feel how heavy and hopeful this responsibility is. Each edit carries consequences beyond the immediate moment. A corrected cell divides. Its instructions spread. Change becomes continuity. This is why the future does not rush. Every step forward is measured. Every success invites more listening, not less. Precision is paired with humility. As the night deepens, you sense the echoes growing quieter in your imagination. Not silenced completely, but softened. The past still speaks, but it no longer shouts. And as this chapter draws to a close, another image begins to form. If such careful edits can ease inherited suffering in humans, what might they do beyond the body, in the quiet fields where life grows and feeds the world? You step away from the human body now, gently and without severing the thread of attention. The same molecular logic that shapes cells also shapes fields, forests, and farms. Life does not separate itself into categories the way humans do. DNA speaks everywhere, using the same alphabet, telling different stories. You imagine standing at the edge of a field just before dawn. The air is cool. The soil is dark and rich with quiet activity. Nothing here looks technological. Leaves rest on stems. Roots press downward. Growth happens slowly, almost invisibly. Plants, like people, inherit instructions. Each seed carries a memory of what came before it. How to grow tall. When to flower. How to respond to drought, heat, or cold. These instructions are not perfect. They are simply what survived. For thousands of years, humans have shaped these stories indirectly. Saving seeds from stronger plants. Crossing varieties. Waiting seasons to see results. This was editing through patience, guided by observation rather than precision. CRISPR enters this long tradition quietly. It does not replace farming wisdom. It refines it. Instead of waiting generations for a helpful change to appear by chance, scientists can now make a small, intentional adjustment. One letter. One instruction. You sense how modest these changes are. A plant that uses water more efficiently. One that resists a particular disease. One that tolerates heat a little better. The plant remains itself. Its taste does not vanish. Its role in the ecosystem stays intact. This is not about forcing nature into unfamiliar shapes. It is about helping plants cope with pressures that are changing faster than evolution alone can handle. Droughts grow longer. Temperatures rise. Pests spread into new regions. You imagine a plant facing stress it was never prepared for. Its instructions, once sufficient, now struggle. CRISPR offers a way to gently adjust those instructions, not to dominate the plant, but to support it. One of the quiet strengths of this approach is what it avoids. Many CRISPR-edited crops do not introduce foreign DNA. They simply adjust what is already there. A sentence rewritten using the same language. You feel how this matters emotionally, not just scientifically. Food is intimate. It enters the body. It carries trust. Changes must feel respectful. Farmers begin to notice differences. Crops that require fewer chemical treatments. Plants that survive unpredictable weather. Fields that feel more stable, less fragile. This stability ripples outward. Healthier plants mean steadier harvests. Steadier harvests mean more reliable food supplies. Hunger is not always caused by lack of food, but by instability. CRISPR, used carefully, can reduce that instability. You imagine a community where crops no longer fail as often, where farmers sleep more easily, where food prices fluctuate less wildly. These outcomes do not feel dramatic. They feel relieving. Of course, caution remains. Ecosystems are complex. A change that helps one crop must not harm others. Scientists test extensively, listening for unintended consequences. Editing plants requires the same humility as editing cells. You notice how this work mirrors the earlier chapters. Guidance before cutting. Repair before change. Listening before action. The rhythm remains consistent. CRISPR does not make farming effortless. It does not remove uncertainty. But it offers another tool, one that works at the level where growth begins. As the night deepens, you sense how quietly radical this is. Feeding more lives not by expanding land endlessly, but by helping existing land work more gently and effectively. You imagine seeds resting in soil, carrying slightly altered instructions. They do not know they have been edited. They simply grow, responding to sunlight and rain as they always have. This is perhaps the most calming realization. Life does not experience editing as intrusion. It experiences it as instruction. Growth continues. And as you breathe slowly, you feel how this chapter widens the story. From cells, to bodies, to fields. The same tools, the same care, now touching the systems that sustain entire populations. You slow your pace now, almost instinctively. Up to this point, the journey has moved through discovery, precision, healing, and growth. Each step has felt careful, even gentle. And yet, here, something changes. The questions grow heavier. The air feels thicker with responsibility. Editing life is not only a technical act. It is a moral one. You sit with that thought, letting it unfold without judgment. The tools themselves are neutral. Proteins do not carry intention. Molecules do not know right from wrong. Meaning enters only when humans decide how, when, and why to use them. This is where science meets choice. For most of history, humans influenced life indirectly. Through breeding, through environment, through selection guided by need and chance. These changes were slow, often reversible, and limited by time. CRISPR changes that rhythm. It compresses generations into moments. It allows intention to move faster than reflection if care is not taken. You feel the tension in that speed. Just because a change is possible does not mean it is wise. This idea repeats itself quietly in ethics discussions, whispered rather than shouted. The ability to edit DNA forces humans to ask what they value, not just what they can achieve. Some questions are easier. Is it right to reduce suffering? To heal disease where possible? Most people feel a quiet yes rise in their chest. Relief, when it is careful and consensual, feels aligned with compassion. Other questions blur at the edges. What counts as disease? Where does healing end and enhancement begin? When does correction become preference? You notice how these questions resist simple answers. They shift depending on culture, history, and personal experience. Ethics, unlike enzymes, does not operate on fixed rules. It listens, adapts, and evolves. You imagine scientists, ethicists, and communities sitting together in quiet rooms. Conversations stretch late into the night. Voices are calm but serious. No one wants to rush. Everyone senses the permanence of these decisions. Editing somatic cells, which affect only one individual, feels more contained. The changes live and end with that body. But editing germline cells, which pass changes to future generations, feels different. The edit becomes a message sent forward in time, without knowing who will receive it. You feel the weight of that unknown. Future lives cannot consent. They inherit decisions made in a different world, with different pressures and values. This awareness slows progress intentionally. Many countries set boundaries. Some edits are allowed. Others are paused. The rules vary, but the intention is similar: to give society time to think before acting irrevocably. You notice how this restraint contrasts with the image of reckless science often imagined in stories. In reality, much of modern biology is defined by hesitation, by layers of review, by pauses built into the system. This slowness is not weakness. It is care. Still, fear enters the conversation. Fear of misuse. Fear of inequality. If editing becomes available only to some, differences could widen. Health could become privilege. Choice could become pressure. You sit with these fears without amplifying them. Fear, like hope, deserves listening. It points to what matters. Ethics asks not only what is possible, but who benefits, who decides, and who bears the risk. These questions are not solved by data alone. They require empathy. You feel how deeply human this chapter is. The same species that learned to listen to cells must now listen to itself, to its values, to its limits. There is also humility here. CRISPR is powerful, but not omniscient. Off-target effects exist. Long-term consequences are not always predictable. Acting cautiously protects not just morality, but biology itself. You imagine ethics as another kind of guide RNA. It does not cut. It directs. It tells power where to pause, where to proceed, and where not to go at all. This guide is imperfect, shaped by debate and disagreement. But its presence matters. Without it, precision tools become blunt instruments. You notice how the story keeps returning to the same rhythm. Guidance before action. Listening before change. Repair before progress. Ethics fits naturally into this pattern. As night deepens, you feel the unresolved nature of these questions. They are not meant to be answered once and forgotten. They require ongoing attention, generation after generation. Editing life means accepting responsibility not just for outcomes, but for intentions. It means staying open to revision, willing to stop if harm outweighs benefit. You breathe slowly, feeling both awe and caution coexist. CRISPR does not force a future. It invites one. The path chosen depends on collective care. And as this chapter settles, you sense another boundary approaching. Even with guidance, even with ethics, precision itself has limits. Tools can slip. Outcomes can vary. Understanding those limits becomes the next act of responsibility. You sit with the idea of ethics and responsibility, letting it settle into a quiet rhythm. But now, your attention drifts back to the molecular world itself, where even the most careful actions can have unexpected outcomes. Precision is real, yes, but it is not absolute. Every system, no matter how refined, carries limits. You imagine the Cas enzyme paired with its guide RNA, drifting through the cell. For the most part, it behaves perfectly. It searches, aligns, and cuts only where it is supposed to. And yet, sometimes, subtle resemblance elsewhere in the DNA can tempt it. A sequence almost identical to the intended target might receive a fleeting visit. A nick here, a change there. Unintended, invisible, yet meaningful. These off-target effects are not dramatic accidents. They are quiet, gentle misreads of the molecular language. The system does not panic. It continues with calm. Yet the consequences can ripple through cells, subtly altering the story written in the code. Life absorbs the difference, sometimes noticing, sometimes not. Scientists have learned to anticipate these slips. They test, measure, and refine. They design guides to minimize risk, check multiple times, and observe the results. And yet, the horizon of certainty remains just out of reach. Perfection is not a destination, but a guiding light. You sense the beauty in this imperfection. Life itself has always tolerated slight errors. Mutations, small mistakes, even the occasional failure of repair are not catastrophic. They are part of the rhythm of adaptation. The cell continues, learning quietly, over and over again. CRISPR mirrors this rhythm. Its actions are precise, but never infallible. And the awareness of these limits encourages humility. Power without understanding becomes recklessness. Knowledge paired with caution becomes wisdom. You imagine the patience required in research. One step at a time. One cell at a time. Sequencing, testing, verifying. Adjusting the guide. Waiting. Each iteration is a lesson. Each discovery builds confidence while reminding you of the unknown. This is where science begins to feel almost like meditation. Observation, reflection, careful action. A constant dialogue between intention and outcome. Mistakes are not feared, they are listened to, integrated into understanding. Progress advances, not through force, but through attentive repetition. You notice how this parallels the natural world. Evolution itself does not achieve perfection, only survival. Slight imperfections persist. Some fade. Some become new features. Life does not demand flawless execution, only continuity and adaptability. As night deepens, you feel this lesson apply beyond DNA. Limits exist in every system, including human knowledge and ambition. Tools, however sophisticated, are guides rather than masters. They extend reach, but never replace judgment. They invite mindfulness, not recklessness. And yet, there is a quiet optimism here. Awareness of limits does not halt exploration. It channels it. It teaches restraint without stopping curiosity. Every careful adjustment, every patient observation, adds to understanding while respecting uncertainty. Even as you rest with the knowledge of off-target effects, you also notice progress. CRISPR continues to improve. Researchers innovate with safer enzymes, refined guides, and more precise delivery methods. Learning continues, gently, continuously, like waves shaping a shoreline. You sense how fragile this balance is. Precision is powerful, yet incomplete. Ambition is necessary, yet must be tempered with care. Knowledge grows alongside responsibility, and each informs the other. You feel the quiet weight of that interdependence. There is trust in the system. Trust in the cells to respond, in scientists to observe, in life itself to adapt. The horizon of perfection may never arrive, but the journey toward it shapes each step with intention. As the chapter draws to a close, you understand that limits are not barriers but guides. They teach patience. They teach humility. They remind you that even when tools are sharp, the story remains larger than the scissor, larger than the guide, larger than the cell itself. And in that calm understanding, another thought begins to form. If limits define what is possible at the molecular scale, what might restraint and guidance teach about shaping the future beyond the lab?